Hi, everyone. I'm Roz Dakin, and I'm excited to tell you today about how my co-authors and I have been examining how hormones can explain variation in animal social networks. Social networks are ubiquitous in animals and humans, and the structure of these networks can affect many important processes, from things like the spread of novel pathogens like the coronavirus down to the evolution of behavior. The challenge that I want to talk about today is understanding how properties of whole social groups are driven by mechanisms at the level of individual animals. We've been studying this question in the wire-tailed manakin, a small bird that lives in the Amazon and has a remarkable social system. Much like humans, male manakins form long-term social partnerships with other males, and they cooperate with each other to gain status. In the wire-tailed manakin, we have dominant territory holders and subordinate floaters. The dominant males invite other visiting males into their territories and perform complex coordinated displays, like the two males you see here. Previous work by my collaborator Brant Ryder and other co-authors has shown that male social behavior is associated with fitness in this species. Young males with more social partners are more likely to ascend to territorial status, and territory holders with more social partners sire more offspring. So it's good to have friends. Recently, we've been studying the dynamics of these social partnerships by using proximity tags to monitor multiple manakin social groups over time. These tags allow us to collect repeated measures of the social structure for each small group within the population. In these diagrams showing just a subset of our data, the nodes represent individual males and the edges represent social partnerships between them. Along with our collaborators Ignacio Moore, Ben Vernasco, and Brent Horton, we found that circulating testosterone can predict variation in male social behavior. As a male ascends in status from floater to becoming a territory holder, his testosterone increases. But once a male is a territory holder, we found that too much testosterone decreases his cooperative behavior. And we've seen this both in correlational data, where the males with the highest average testosterone score are the least social, and we've seen it in an observation where we've manipulated male testosterone levels, and we've shown that the males that receive a testosterone implant have a significant decrease in their cooperative behavior that exceeds that found in a control group. So we know in this system that testosterone influences an individual male's behavior. And as further background, we found that one male's social behavior is associated with the recent behavior of his close partners. And most recently, we've shown that there's considerable variation from one group to the next in group level properties of these social interaction networks. And so this sets the stage to now ask whether hormones can explain variation at the group level. We were particularly interested in group level properties that are important for cooperation. The first property we examined is called social specialization. This parameter captures the exclusivity of the relationships between partners in the network. And in these Sankey diagrams, each of the rectangles in the vertical columns represents an individual manakin, and the highways between them represent social interaction frequencies between dyads. So we have manakin networks that are highly specialized and some that are unspecialized. A highly specialized social network is expected to improve familiarity and behavioral coordination among partners, which is good for cooperation. The second property we examined is called network stability. This captures the extent to which social relationships persist from one time point to the next. Greater temporal stability of partnerships is also expected to increase the familiarity and reciprocity within a social network. As the predictor variable for these two properties, we considered something that we're calling collective testosterone. And we get this measure by taking the average testosterone level of the males within a social network, weighted by their social interaction frequencies over a particular recording session. The image in the middle of the slide here shows a manakin group with moderate collective testosterone. And on the left, we have one with relatively low collective testosterone, and in the right, one with relatively high. So the question is, does collective testosterone explain the social specialization and stability of the group? So here's what we found. We found that social networks that were composed of more high testosterone dominant males were less specialized and less stable. And we verified using some sensitivity analyses that these results were robust to a wide range of parameters that you could choose when defining the social network metrics. So hormones can explain the structure and dynamics of whole groups. When a male has elevated testosterone, he may behave in ways that weaken his immediate social partnerships with other males, causing his previous partners to prospect elsewhere and degrading the stability and specialization of the network as a whole. This is a breakdown that could impede the evolution of cooperation in the long term. We expect that steroid hormones may be drivers of social network properties in many other systems, given the important role of these hormones in social behavior across vertebrates. And I want to end by thanking my co-authors and our many sources of support and the organizers of the Manakin Genomics RCN, and by saying that I have opportunities for students to come and join my lab in Canada. In particular, I'm seeking future PhD students, so please do see my website to get in touch. Thank you.